Greetings, students. So population genetics is the study of gene frequencies in a population. So by gene frequencies, what we're talking about is that example from the last screencast where if we say we got about 75% green beetle, 25% brown beetle. Those percentages are basically saying how common genes are in a population. So in our last screencast, we sort of talked about how Darwin was very focused on the broader scale evolution, what we call macro evolution. Here we're going to zoom in on this microevolution, this idea of gene frequencies changing over time. So some background terminology here. First of all, the gene pool. The gene pool refers to all of the alleles in a population. So if you've got a whole bunch of pigs in a particular environment, and some are homozygous dominant, and others are homozygous recessive, and others are heterozygous, imagine taking all those big B, little b's and just dumping them in a pile. That pile would represent the gene pool. It's all of the alleles that exist in that population at any given moment. The allele frequency then is the ratio of a particular allele to the total number of alleles in a population. And I think you're probably pretty familiar with this from math, but just in case. So let's suppose you had, this is an incomplete dominance example where you've got red flowers, pink flowers, and white flowers, and I said calculate the frequency of CRW. How would you do that? Well, let's go to the dot cam here, and lo and behold, there's that same problem. So if I said calculate the frequency of CR and CW, so for CR, there were 320 red flowers in that population, and each red flower is homozygous for this allele, right? So there's one here, two here. So each of these 320 has 2 copies of that CR allele. So we would say, all right, there's 2 alleles times 320 individuals. So that's going to be 640 copies of that particular allele. Now, over here, though, the pink individuals have 1 copy of that CR. And so we'd say, all right, 1 times 160 individuals is going to be 160. And then we would simply add those up, and we would get 800. So we would say there are 800 CR alleles in this population. Now, in order to finish the problem, though, it's asking for a frequency as a percentage. So we need to divide 800 by the total number of alleles. Well, if you count the number of individuals, 320 plus 160 is 480, plus 20 is 500, and each of those 500 individuals have 2 alleles, right? So there's 1,000 alleles total in this population. So the frequency of the C big R would be 0.8. That would be the frequency. We could repeat that analysis for CW, and it may not be a terrible idea for you to just pause the video and see if you can do it on your own. But I'm going to go ahead and do it a little bit more quickly. So CW, we got 20 individuals who each have 2 copies, so 2 times 20, plus, and I should have added a plus here, plus 160 individuals who have 1 copy, so 1 times 160. And so that's going to equal 40 plus 160, which is going to be 200. And then we take 200 over the total number of alleles in the population, which is still 1,000, and we get 0.2. And that should not be a surprise to you, because if 80% of the alleles are the C big R, then the other 20% should be the CW. So, so that's how you can calculate the frequency of an allele in a population. And here's just some math to kind of help you understand that a little bit more. So since the definition of evolution is changes in allele frequencies within a population over time, we need to be able to work with these gene frequencies mathematically so we can track those changes. So time for some math, children. Time for some math. The central concept within population genetics is something called the Hardy-Weinberg equation. This describes the allele frequencies of a non-evolving population. So if there's no evolution happening, we would say that a population is at Hardy-Weinberg equilibrium. I keep saying Hardy-Weinberg, that's weird. Hardy-Weinberg equilibrium if we're not evolving. So think of Hardy-Weinberg equilibrium as a null hypothesis. It describes a situation where really nothing interesting is going on. It's just, gene frequencies are staying constant all the time. So, by comparing actual populations to what we predict with the Hardy-Weinberg equation, then we can kind of see if something is happening. So, basically, yeah, let's just talk about this example here. So here's two sets of data, population A, population B. Here's the frequency of big A, generation 1 through 4, population of little a, and then here's the same for population B. Take a moment. Which population is evolving? And hopefully you said, oh, it must be population B because the gene frequencies are changing over time. We can see that the frequency of little a is getting less and the frequency of big A is getting more. Why that's happening, we don't know, but it's clear that they're changing. Next, which remains at Hardy-Weinberg equilibrium over all 4 generations? Well, that would be population A because nothing is happening to the gene frequencies over time. Big A is just as common in the first generation as in their great-grandchildren. Great-great-grandchildren? Great-grandchildren. I don't know. So this one's at equilibrium, this one's evolving. That's kind of in a nutshell how this works. So now we get mathy. And note that these equations we're about to go through are in your AP Biology formulas and equation sheet. So you want to pull that up. If you don't, it's in Schoology, somewhere in that white administrative nuts and bolts folder. But here are these two equations. P squared plus 2pq plus q squared is equal to 1. P plus q is equal to 1. And P is the frequency of the dominant allele in a population. Q is the frequency of the recessive allele in the population. So, there's our equations. Write those down for sure. And then here's our variables. P is the dominant, Q is the recessive. So, this should, by the way, make sense to you. Like, these are just kind of like, oh, duh, if you think about it. Because P plus Q equals 1, for example, if one allele is dominant and the other allele is recessive, then the frequency of the dominant plus the frequency of the recessive should represent 100% of the alleles, right? So all the dominant alleles plus all the recessive alleles together should be 100% of the alleles. So that equation just kind of makes sense. This equation is a little bit more complicated. These equations actually represent the 3 possible genotypes. So P squared is actually the frequency of the heterozygous dominant. 2pq is the frequency of the heterozygotes. And q squared is the frequency of the homozygous recessives. And so all this equation is saying is if you take all the homozygous dominants, all the heterozygotes, and all the homozygous recessives, again, that should account for 100% of the alleles. So both of these equations are just kind of stating the obvious, really. And if what I just said doesn't make sense, then here's a little bit more on why that would be. So I'm going to go ahead and switch over to my DocCam. And if the probability of A is equal to P, then the probability of big A, what is that going to equal? Well, the probability of 1A is P. So the probability of a second A is also P. And so the probability of having a big A and a big A is going to be P and P. And remember the and rule from simple probability. P and P means we multiply. And so it's going to be P squared. And you can repeat that for the others. So the probability of little a is going to be Q and Q, which is going to equal Q squared. And then for big A, little a, there's actually 2 ways. You could have a P and a Q, or, so that's the plus, oops, you can't even see that, or, so P and Q, or it could be the other way. It could be the little a and the big A. And so that is going to equal 2pq. So those terms sort of derive themselves based on some pretty fundamental concepts. And you can even do like a Punnett square kind of deal. So if you have the two heterozygotes, PQ cross PQ, P squared, PQ and PQ and another PQ and then Q squared. So anyway, that's where they come from. So let's use these equations to try and solve some problems. So problem number one, if P is equal to 0.6, find Q. And so I've actually written these problems down on a piece of paper, and we can just use the .cam to go through them. So if P is equal to 6, find Q. Well, let me use this purple. We know that P plus Q is equal to 1. So if P is equal to, oh shoot, that should be 0.6. So if P is equal to 0.6, simple algebra tells us that Q is equal to 0.4. Okay, that's not too bad. How about this one? If P is equal to 0.25, what is the frequency of the heterozygotes? Well, rule of thumb is you always want to solve for P and Q. So if P is equal to 0.25, Q is going to be equal to 0.75, and based on that equation, 0.25 plus 0.75 equals 1. So I know that Q is equal to 0.75. Now the frequency of my heterozygotes is going to be 2PQ, based on what we just discussed, and that's going to be 2 times 0.25 times 0.75. And that is going to equal a number, and that number is, I'm not even sure what that number is. Hopefully it's 0.375. So there's answers on the slideshow there. So if you do the math, I'm going to trust myself that is 0.375. So that's the frequency of the heterozygotes. The next question, in a population of 1,000 individuals, 90 expressed the recessive phenotype. How many homozygous dominant individuals are there? Ugh. Okay, well this is getting a little more complicated. Let's take this one out. So, population of 1,000 individuals, 90 express the recessive phenotype. Now note that it's saying phenotype. So if they're expressing the recessive phenotype, that means they must be little a, right? That's the only way you express the recessive phenotype. And little a we know is Q squared. So in a population of 1,000, 90 express this. So that means that Q squared is going to equal 90 over 1,000. 90 over 1,000. And so to solve for Q, all you need to do is take the square root of each side of the equation. And 90, boy, I should have grabbed a calculator. I didn't think about it. 90 over 1,000 is going to be, well, shoot. Pause. Well, gosh, that wasn't that hard, actually. So let's go back to our .cam here. 90, so 90 over 1,000 is basically going to be 0.09. And so the square root of 0.09 means that Q is equal to 0.3. Okay? So if Q is equal to 0.3, that immediately tells me that P is equal to 0.7 because P plus Q should equal 1. So now that I have P and Q, these are important. Now it's asking how many homozygous dominant individuals are there. Well, to solve that, what I'm going to do, oh, that gets rid of that, that's nice. What I'm going to do is homozygous dominant is going to be big A, which is equal to P squared, which is equal to, since I just calculated P is 0.7, 0.7 squared. And if I plug 0.7 times 0.7 into my calculator, I get 0.49. I'm not sure how seamless this editing is, but I messed up that last one. So here we go. So we're at 0.49. And so then we go back and we say, okay, if there's 1,000 individuals in the population, 49%. So 0.49 times 1,000 individuals is going to equal 490 individuals. So 490 individuals are going to be homozygous dominant. So this slide in the slideshow sort of summarizes the correct answer with a little bit of kind of focus on what you're trying to solve and why we went and solved it the way we did. So all of those answers match mine, mercifully. And so then there's one last problem. This is a little bit of a tricky one. What if we said instead of 90 express the recessive phenotype, we say they express the dominant? We'll look at that in our next screencast.